Okay, go ahead.
All right. Well, it's my pleasure to be here today. I am Irene Gregory-Eaves. I'm a professor of biology at McGill University, and I've had a long-standing relationship with the alms going back to my first student who did some field work, I believe in 2006 with alms teams. And so, yeah, I have a lot of respect for this organization. And today I'm going to talk about the first Canada-wide assessment of lakes. This was a standardized sampling of lakes, the first time it was done nationwide. And I want to, address what are we learning? We have been producing papers and students have been working on this data set, but it's really exciting that this data set was just released to the public this summer. So any one of you can go and download the data through the Gordon Foundation data portal and find your lake and compares it to other lakes, as well as there is the Lake Pulse, www.lakepulse.ca has its own comparative portal. If you don't want to do your own analysis and you just want to know how your lake compares to others in the region, you can check out this data portal. Okay, so I should say, though, that this project was a massive team effort led by Yannick Huot, who's a professor at the University of Sherbrooke, and there was PIs from across Canada involved in this. So this is where it started, with a bunch of PIs getting together and writing a grant, but certainly not where it stopped, because a lot of the people who were out in the field collecting samples over 3 summers are the students and postdocs. And so this was my group, many of whom were involved in the Lake Pulse project. And then in addition to the PIs and students and postdocs, we drew in outside collaborators as expertise were needed, popped up. So really, this was like a cross-Canada limnology effort. Many people aboard this ship, if you will. And I don't think I need to go into great detail and excite this audience about the importance of lakes, but I do like this paper by Williamson and colleagues who argue that lakes are great sentinels, integrators, and regulators. There are many species that live within lakes that are very sensitive to changes that happen and give us an early warning indication that things are changing. They're great regulators because there's lots of geochemistry, a lot of chemical cycling that happens within lakes. And then finally, they're great integrators, that many lakes have a faithful temporal record in their sediments. And this allows us to go back in time and reconstruct how the lake, the airshed, and the watershed has changed over time. So it's interesting that up until now, limnology has always been very strong in Canada, but has been very regionally focused. And it seemed much overdue for us to have a national sampling program because there are more lakes in Canada than anywhere else in the world. And so this is a nice map put together by colleagues of mine at McGill that show the density of lakes across different areas. And where we have lots of lakes is largely due to where glaciers sat 20,000 years ago and carved up the landscape and left lots of holes in the landscape, if you will. So the goal of Lake Pulse was multifaceted, but a key component of it was what we call the large-scale sampling of lakes, going across the country and sampling lakes in a very standardized way, and doing so in a very strategic fashion, which I'll talk about in a moment. So there were some in-situ moorings collecting high-frequency data. There was a large-scale sampling of lakes that was a one-time sampling midsummer. At each of these lakes, we also took cores. I'll talk a little bit about that. We also tried to pull together existing data sets and then use scenarios, particularly climate forecasts and hindcasts, to get a sense of where we might be going in the future. And there was also a large remote sensing arm. So the network itself was broken into 4 different themes. The first theme really asked the question, where, by how much, and why have Canadian lakes changed during the Anthropocene, this period of heightened human activity? Another major theme was really focused on indicators. How do taxonomic, molecular, and biochemical features of the plankton, benthos, and microbes reflect lake alterations, and which ones might be the best indicating health of Canadian lakes? A third theme was looking at optical, morphological, and watershed properties, so that although we as a group could only sample 664 lakes, that we could then scale up this to most lakes on the landscape. And then finally, a sort of forward-looking theme and one linked to ecosystem services. So as I said, there was 17 PIs across the country with many students and postdocs, and lots of exciting stuff is reported on the Lake Pulse website. And I'm just going to talk to you about some of the studies my group was involved with in Theme 1 and Theme 2. And germane to Theme 1, a big part of it was collecting cores. As I mentioned, lake sediments are natural archives of how the lake, its watershed and airshed have changed over time. And it would have been a really amazing, awesome, but buried us in, many, many careers to get through full cores from all of these lakes. So what we did is we collected cores from each lake, but in each lake we took a surface sample to reflect recent conditions and a bottom sample to reflect pre-industrial conditions. And we did a big literature exercise to identify how deep did we need to go to get ideally pre-1880 conditions. Then we analyzed these top bottom samples, as we're calling them. Sometimes not all of the samples, because that would have been 664 times 2. That's a lot of samples to process. So often we did, in terms of analyses, a subset, but we tried to keep the block strategic design, which I'll talk about in a moment. And then we have done full cores on a subset again, usually on the day before shipping. So if we consider a week of sampling, the 6th day was the last day of sampling, and then on day 7 was part of it was to ship those samples back to Sherbrooke, where they were then distributed across all the labs, as well as to have a bit of a day off. So on the 6th day, we took a full core, and that got shipped to Sherbrooke. So we are looking at full cores from a subset of lakes as well. I'm not going to talk so much about that today, though. So by full core, I mean you're looking at the whole profile. So first, we had to figure out, well, which lakes to sample? How are we going to do this? And we really did want to model as much as possible our work off of the US National Lake Assessment Program, which has had several iterations now, but they had another sampling in 2017, which matched our first year of sampling. That year we went to Eastern Canada. So they have a minimum size of 0.04 kilometers squared. We went to 0.1 kilometers squared to 100 kilometers squared. So 10 to 10,000 hectares. That was the size of lakes that we sampled. The big lakes we figured in Canada actually are pretty well sampled already, such as the Great Lakes, and very small shallow lakes could well be dry if we sample only in midsummer, so we excluded them. So we sampled what you might say is the Goldilocks zone. Then we also did a stratified random sampling design. We first wanted to identify what was the major land uses in each watershed. And for this, we had to figure out, delineate all the watersheds within 1 kilometer of a road in Canada in our 12 ecozones that we were studying, and calculate what we're calling the human impact index, which is if you rank different land uses according to the intensity and then take the sum score at the watershed scale, you can categorize them into high, moderate, and low intensities. And then lakes are of different sizes as well. So we had different bins. So with these different bins, we did what we call stratified random sampling, so that we had roughly equal numbers of lakes in each of these squares. So we did this in 9 core ecozones, and then the 3 northern ecozones that we hit, we did more modest sampling, always using the stratified random sampling design, and then added a few additional lakes that were of importance to partners. So what does this look like on the map? This is a map that was coming out in a publication in the Canadian Journal of Fisheries and Aquatic Sciences, which shows the ecozones that we sampled. It shows all of our sites shown as dots across the country, and each lake is color-coded according to a human impact score. So here we can find ourselves in Alberta here, where there is quite a bit of high human impact due to the presence of widespread agriculture. So this is what it looks like across Canada on a map. But of course, you know, this is what we might see on the landscape. And I believe this set of photos is from British Columbia, but a similar set of photos could easily have been taken in Alberta, where you have a wide range of different habitat types. So some lakes close to the alpine, some maybe more affected by cyanobacteria, and a variety of different land uses. At each lake, we measured many variables, essentially as many as you could in a single day with a team of 4. And all samples were collected between late June and early September, mostly between July 1st and August 31st. And this was done intentionally to minimize seasonal effects. If the lake was going to be stratified, if over like a stratified summer, it would have been stratified by this point, okay? So we collected physical, chemical, biological, and optical measures. The optical measures were important so we could have in situ data to calibrate with remote sensing. And the overall goal, why this network was funded, was to look at lake ecosystem health, which, you know, even when we started writing this grant, we debated what is ecosystem health. And there are references that we can use in the literature, but I think a general common understanding is that health can be defined in a comparative way. So you might compare at one lake to a reference lake in your ecozone. So one that has a lower human impact in terms of land use in the watershed. Or in the case of paleo or long-term data, you could refer to a pre-disturbance condition. And then in terms of what we measured, and what I'm going to present to you today are analyses of water chemistry. We've looked at algae and diatom assemblages in sediment cores. We've looked at cyanobacteria in the water column and toxin concentrations. The blue bullets are ones that we measured in the water column. The brown bullets are things that we measured in sediment cores. Our group was also involved in studies of zooplankton, both in the water column and in the sediments, as well as sediment geochemistry, but I just don't have enough time to talk to you about that today. Okay, so let's launch in and look at the water chemistry. This was work that was done by a student doing an undergrad project. He was able to look at the basic data, looking at water quality parameters. And here what we've done is constructed what's called a principal component analysis, which is an exploratory approach to visualize relationships amongst different variables. In this case, water quality is shown in black variables, and then we've plotted passively land use variables. And so here you can see that sites that have more agriculture and more urban and more pasture tend to be those lakes that have higher concentrations of ions and higher concentrations of nutrients. And then on this plot here, just so things are not too crowded, we're showing all the sites. And the sites are color-coded by ecozone. So if we dig into this a little bit more, we can zoom in and look at this first axis. So we've extracted the first axis scores for each of the ecozones across the country. And we can see that the prairies and boreal plains have a much lower or negative PC1 score relative to the other lakes, which means that they have, on average, higher nutrients and higher ions than the rest of the country. Well, these are also ecozones that have extensive agriculture. So to probe this a little bit further, we then looked at a handful of ecozones to see, well, are nutrients in fact related to the proportion of agriculture in the watershed? And so that's what we're showing here across 4 different ecozones: the boreal shield, the mixedwood plains, the boreal plains, and the prairies. And admittedly, there's in some cases decent relationships with agriculture. For instance, here, the mixedwood plains explaining 40% of the relationship between total nitrogen and agriculture, and in other areas, there's no relationship. Another thing you can take away from this graph, or this series of graphs, is the intercept. The intercept tells you, it can act as a reference guideline. So in the absence of agriculture, what would this region's likely phosphorus or nitrogen be? And you can see that that varies across different regions. So the prairies has a much higher baseline than the mixedwood plains, for example. So naturally, one would expect that the prairies, even in the absence of agriculture, have higher nutrients. And I think this is something that we probably knew, but it's nice to see demonstrated in a quantitative way. And in some ways, this explains why there is as much agriculture in the prairies as there is today. Farmers are smart and figure this out, and this is where the soils are quite rich. And if we go back far enough in the geological past, we know that this was an area of the Western Interior Seaway. And so this is where rich soils develop, because essentially they're old marine deposits there. And the soils are quite naturally nutrient rich. So there's some explanation for this. But we also wanted to know, now in the presence of agriculture, how much have things changed? And to do this, one really needs to collect cores. So we did, in fact, do that. And so here now we're using both diatoms, which are unicellular algae. Their shells, if you will, have species-specific markings on them. So from the what we call subfossils, or the shells alone, we can tell how the community has changed. And we know that certain ones are more indicative of high nutrients versus low nutrients. And we can quantify in a single metric how much change we've observed by calculating what's called dissimilarity. So it looks at the assemblage in the bottom and the top and quantifies how much change we've seen. So we've done this across many different ecozones. First, we'll look at the Pacific Maritimes here, and we can see that the Bray Curtis across the, I would guess, I'm sorry, I'm not guessing, but from what I remember, there was about 20 lakes in this area that we did this study on, and there's some range, but roughly around 0.5 dissimilarity. So it hasn't seen the greatest amount of change, it hasn't seen no change. And then if we look across all the ecozones, we can see that there are differences across ecozones. So the prairies, for example, is the ecozone, along with the mixedwood plains, that has changed the most. So not only did the prairies have a higher nutrient-rich background condition, but it also has changed. And we know from looking across lakes that we've now been by high impact versus low impact land use in the watershed, that there is a significant difference. So although it started quite nutrient rich through human activities, we have definitely seen a greater change in this area based on the diatom assemblage. If we look at the sediment chlorophyll A, we're looking at chlorophyll A and its breakdown products. And to do this, we're using a visible reflectance spectroscopy. In this graph, here we're looking at the sediment chlorophyll A in the bottom, that deeper sample, that pre-industrial sample, versus what we see in the top. And in black is the 1-to-1 line. So if all lakes showed no change, they would all line up against the 1-to-1 line. But what we actually see is that 80% of lakes have shown an increase. And this is true across all ecozones. This is really a widespread signal. And we also found by looking at the species composition of the diatoms that there was also a widespread increase in the number of planktonic, or the proportion of planktonic diatoms in the modern sediments. Both of these indicate likely that it had to be a more widespread factor, of which climate change is the most likely contender. And regional studies where they have continuous core data looking at the proportion of planktonic diatoms and been able to compare that to climate records have found indeed that that is the metric that explains most of the variation. So closing that chapter and moving on to the cyanobacteria, I'm sure, as you know, cyanobacteria are a common symptom of eutrophic conditions, cyanobacteria blooms, and things seem to be getting worse with climate change. So this was work done by a PhD student who analyzed all of the data from our one-time sampling of lakes across Canada. What does the cyanobacteria biomass midsummer look like? And so we can definitely see that there are some hot spots, again, in the prairies and boreal plains, but also in the mixedwood plains in eastern Canada, as shown here. And the top 4 taxa that were detected were Aphanizomenon dolichospermum, Microcystis. The top 3, of course, are bloom-forming taxa, and then Aphanocapsa is a picocyanobacteria, so a very small cyanobacteria. And Paul did quite a lot of modeling work, was able to build a pretty strong model at this scale, so was able to explain 55% of the variation, and phosphorus was indeed the strongest variable. So we knew that nutrients were important from smaller studies, but now we're seeing this at the national scale. In addition, some other variables were indeed important. And we also measured microcystins using both an ELISA kit as well as looking at the different congeners of microcystins. Here we're just looking at the findings from the ELISA kit, and here you can again see a hot spot in the prairies and boreal plains. But on average, nationally, the picture is good, that we generally have very low concentrations of microcystis. Again, he did a modeling exercise. You have to model presence-absence data separately from when the microcystins are present. But when microcystins are present, we can explain 70% of the variation, and actually microcystis biomass was the strongest predictor, in addition to nutrients and temperature. So that wraps up the Lake Pulse portion. What I want to just highlight now in closing is a recent study by some colleagues, Irene and Charlie Trick, and their student, Kevin Erat. They took cores throughout Alberta, Saskatchewan, and Manitoba, and you can see on the map where those sites are. And in each of these cores, they measured chlorophyll A using the same approach that I just talked about. And they also calculated the human impact index in each watershed, just as I talked about. And for each core, they looked at where is there a rate of change in the trajectory. So for chlorophyll A, they found that the change point was actually the earliest in their high impact lakes. So on average 1945, whereas the low impact lakes, those on average didn't change until much later, the late 80s. They also measured cyanobacteria using paleogenetics and found roughly the same thing. Again, similar timing. And then finally, they measured microcystins. And here you can see that they found no change amongst the different human impact categories for their lakes. So it's interesting, this dichotomy of chlorophyll A and cyanobacteria showing a strong association with land use, although also partially explained by climate, whereas microcystin, really here they're finding climate is the main driver. And the link for their paper is right here. You can go check out for more details. So I would just like to wrap up by concluding that we've talked about water chemistry and diatom assemblages from the sediments, and this shows clear evidence of the impact of land use on lakes downstream, the widespread increase in chlorophyll A, as well as the relative increase in plankton, are best explained by climate changes over the last 150 years through warmer conditions and a longer growing season. With the cyanobacteria sampling lakes across Canada, we show that nutrients are still the most important predictor of cyanobacteria biomass. But using this snapshot approach, we do leave some variation unexplained, up to 45%. And finally, with the microcystins, we found that microcystis biomass was the strongest predictor of microcystins. And the sediment core work by colleagues has shown that land use and climate change are important for both chlorophyll A and cyanobacteria, but climate seemed to be more important for microcystins. And so with that, I'd like to thank you for your attention and thank the many students, staff, partners, and funding agencies all involved in making this possible. Thanks very much, and I'm happy to take questions.